All right, so this is chapter 3, talking about basic Mendelian genetics. And it starts with our chapter opener showing this young lady with red hair. And the opening story is about the genetics of red hair. That's always pretty interesting. And apparently, there's been some controversy over whether it's dominant or recessive. Sometimes it appears to be a dominant inheritance pattern, sometimes a recessive inheritance pattern. And but it turns out that it is due to one gene, and if you have one mutation in that gene, sometimes you'll get red hair, and so it's dominant in those cases. And then in other cases, it requires two mutations in that gene. So the gene is called melano. Trying to remember what the name is. Melanocortin 1. So melanocortin 1, or the MCR1 gene. And if you have a mutation in that gene, in particular if you have two, you get red hair. So sometimes it's dominant and sometimes it's recessive. That would sort of make sense. So if you want to read more about that, it's kind of interesting. Read the opening story. I'm certainly not going to put it on a test or anything like that. So basic Mendelian genetics, or classical genetics, goes back to Mendel, back in the year of 1865 or so, back in those days. If you ever see a picture of Mendel, it makes you think he was working in like the 1400s or something, but it was back in the mid-1800s, so things like the Civil War are going on at that time, probably just finishing up, and so it kind of puts that sort of time frame. And so he was using P's, and his big advance was that he used numbers to sort of get an idea and come up with hypotheses of what was actually going on. This is just an example of some of the traits that he used, and so we'll use some examples of these. Yellow versus green, round versus wrinkled. I've never used gray versus white. Yellow versus green pod color versus seed color. A lot of different traits that he looked at. And we'll just use a couple of these as examples here and there. And it's always good to go back to Mendel just to get things started out. These are some terms that are important to know. You should all know these already, actually. If you haven't heard of a gene or an allele, you didn't pay a whole lot of attention in Bio 195. So everybody's had some genetics in high school, in Bio 195, but we use these genes a lot, or these terms a lot. So gene, that's an inherited factor. We now know that a gene is a segment of DNA that is inherited, and we'll go into the molecular details of what a gene is. An allele is one or more alternate forms of a gene, and a locus is the place on a chromosome where a gene resides. So if we draw a chromosome, something like this, and there's a spot or a location on the chromosome where the gene lives, and then we got another chromosome, and so this would be homologous chromosomes, or we'll call them homologs. That means one is from dad, that's the paternal chromosome. One is from mom, that's the maternal chromosome. This is the locus, so where the position on the chromosome where the gene resides is called the locus. And each locus has a gene, and the version of that gene is called an allele. So big A or little a is what we mean by allele. And gene is sort of a generic term meaning the DNA that is at this spot. So we'll take a look at that. The genotype, you can have, because you have two possible alleles for any particular gene, a genotype in this case would look like this. That would be a heterozygote. You could be homozygous recessive. You could be homozygous dominant. And then you get a phenotype, or trait is the outward appearance of the particular genotype, and the character is the particular. So the phenotype, let's say, so if we're talking about seeds, we can have round or wrinkled, and so that would be the phenotype, either one of these. The trait, or the characteristic, or the character, is seed shape. I usually, we don't use these words a whole heck of a lot. We usually just use phenotype. I usually use trait, meaning sort of the seed shape, and round or wrinkled being the specific phenotype. So what did Mendel do? He first set up some monohybrid crosses, and he came up with these rules for inheritance that I think now we sort of take for granted, but formally they separate them into all these different separate ideas, but it's kind of all the same thing. So each trait or each character is encoded by 2 genetic factors. All right, so alleles, and one of the big things. And one's dominant and one's recessive. So if we have a heterozygote, then these guys segregate away from each other. And we call that segregation of alleles. So one's dominant, one's recessive, and the alleles separate with equal probability into gametes. So if we have big A, little a, we can get big A or little a, and we can get these in equal probability. Half the time we'll get big A, half the time we'll get little a, but this is in gametes. And how do we get that? Meiosis. So that's kind of what this chapter is about, at least for one gene. So that's a monohybrid cross. And a monohybrid cross is something very specific. It's actually, we'll come back to the details of that one. We've got a little bit more room on the screen. So that's Mendel's first laws, the principles of segregation, that big A and little a segregate away from each other. And then dominance and recessive have sort of specific terms. If you're heterozygous and you see a particular phenotype, we like to say that's the A phenotype. If the A phenotype shows up, that means, so let's say it's for round or wrinkled. Let's say you're heterozygous for the round allele, and it's round. That means this is dominant. And in order to see the wrinkled phenotype, you need to have 2 of the little r's, and that is the definition of recessive. So you need to be homozygous for an allele in order to see the trait, then it's recessive. Pretty simple, but it comes in handy that because that's the definition, if you know that something's recessive and you see the phenotype, then you know the genotype. If you know something is dominant and you see the phenotype, you don't necessarily know what the genotype is yet, because it could be one of these 2 things. So if we look at a monohybrid cross, we always get the same thing. Big A, little a, cross to big A, little a, and we're going to get 3 different potential outcomes. There's 2 ways to get this, and so we get a 1 to 2 to 1 ratio. You can show that in a Punnett square. Mendel did not use Punnett squares. They were developed by a guy by the name of, you guessed it, Punnett. His name's R.C. Punnett. So these are gametes. These are gametes. These are gametes. And then when they fuse, they can give you these offspring. And they look like that. 1's like that, 2's like that, 1's like that, 1 to 2 to 1. Then 3/4 of them will have the A phenotype, because they can have a big A or a little a, and so we indicate that with that little blank space. And there's one way to get 2 little a's. So these probabilities play a big role in thinking about genetics. And then the chromosomes describe, once we start looking at the chromosomes or thinking about the chromosomes, they explain how the alleles segregate. So if we go back, this mentions that Sutton, it was actually Sutton and Bovary sort of separately, came up with the chromosomal theory of inheritance. And what they did was they looked at chromosomes under the microscope, and they saw that there were these things that seemed to segregate away from each other during meiosis in the same way that alleles seem to segregate away from each other in a genetic cross. So they just made the connection. They just said, well, it kind of looks like these things called chromosomes are behaving the same way that alleles do. Maybe they have something to do with each other. Maybe these alleles are carried on chromosomes. And so if we look at chromosomes, they sort of look like this. We can draw them in different ways. And this is just for one gene. So we're just showing one chromosome. And so we've got, I think I'm pressing down a little too hard. So if we got a chromosome, sometimes I draw them like this. And we've got a big R. And then we've got over here a little r. Okay, so that's simple enough. So the genotype is big R, little r. Here, we've done what we call S phase in the cell cycle. We'll look at the cell cycle a little bit more in chapter 2, but S phase is when the chromosomes replicate, and so now they sort of look like this. Sort of. That's how I like to draw them. Sort of. Sometimes I can draw them better. And we got a big R there, little r there, little r there. So, just like this. But the key here is that when we copy these chromatids, the chromatids are then identical because we just made a copy of them. So if we start out with a big R on one chromatid, now we have a big R on the other chromatid. Here we have a little r on this chromatid, and we have a little r on this chromatid. Oh, crossing over. We're not going to talk about crossing over. All right, so don't worry about that. We're gonna come back to that later. I think right now it just confuses the issue. So what happens is chromosomes line up like this during meiosis, and then when they segregate, the big R and the little r go away from each other, and then there's a second round of division in meiosis where big R goes to one gamete and little r goes to other gametes. Because we have two divisions, we actually end up with two of these and two of these. But we like to think about it as one half are big R and one half are little r. We don't really care that there are two of each. We just care about the ratio, that one half are big R and one half are little r. What's really important, so this is, right here, this is metaphase 1 of meiosis. This is anaphase 1 of meiosis, where they separate. And then we've got telophase and cytokinesis and all that. But what we really care most about is at metaphase of meiosis, the chromosomes, these are homologous chromosomes, line up in pairs. And so then they have to be separated away from each other. So that's how the chromosomes account for Mendel's law of segregation of alleles. It's relatively simple when you just have one chromosome, or one pair of chromosomes, and it'll get a little bit more complicated when we have two. So this is just a close-up. Maybe I can get another opportunity at drawing chromosomes. So in here, there's lots of different ways to draw chromosomes. I draw them like this because that's kind of how they look like under the microscope. You can also just kind of draw them like that, and then that ends up becoming sort of like an X. You can draw them like this, where you have one chromatid, a centromere, and another chromatid. So lots of different ways to draw chromosomes. Then when they replicate, let's see, there's one chromatid with an R on it. It replicates, and when it replicates, it makes another sister chromatid. So if it just made a copy of that, then we have to put that on there. And then the other one will have a little r on it. And they line up in pairs. So then so this is metaphase 1, and then during anaphase they'll segregate, and you start out with them look like this, and then eventually they all end up just in their one cell, just like we saw on the last slide. So just like this, so don't worry about crossing over, and then they divide again. So what we've got is 1/2 big R, 1/2 little r. So if we do it Punnett square, and we have two parents like this, 1/2 gives us big R, 1/2 gives us little r, the other parent. One gives us big R, the other gives us little r. That's if we did that would be the results of a monohybrid cross. If we did two of those. So Punnett squares are okay. So here we go. We've got a tall plant, short plant. So this is actually jumping ahead a little bit. And so what this is where this is a test cross. And so what Mendel did was he took a monohybrid cross. He saw a 1 to 2 to 1 ratio. He hypothesized based on that ratio that the alleles were behaving in this fashion. So then to test that, he took some of his tall guys and he crossed them to some of these short guys and asked what happened. And two things could happen. You could have big T cross a little t, and if that's the case, you would get big T, and they would be all tall. The other possibility is you could have big T cross to little t, and you would get one half look like this and one half look like this if these alleles are segregating away from each other. And so he did that, and sometimes he saw this, sometimes he got this result, and sometimes he got this result. And he actually got this result 2 times as much as he got this result. So what that told him was that when he started with these, 2 of them, 2/3 of the ones that showed the dominant trait, 2/3 of the tall plants were actually heterozygous and behaved this way in a test cross. So this is always what a test cross looks like. You always have something, in this case either big T or little t, and so we could say we don't really know what this is. Even if we did know what it is, we could still call this a test cross. A test cross is if we're crossing it to something that is homozygous recessive for the gene we're talking about. And we call this person, or plant in this case, the tester. And then we can do the cross and we can fill in this information. There's a close-up of it. Alright, so these little concept check things, sometimes I keep them in, sometimes I don't. But it's nice to give you just an idea of what some of these simple problems will look like. If an F1 plant depicted in figure 3.4 is back-crossed to the parent with round seeds. Alright, so let's go back. 3.4. Alright, what happened to 3.4? Just want to make sure we've got the right cross. So I happen to have my book here, so I'm just going to look it up and look up 3.4. Alright, so the cross in 3.4 is, so we go back to figure 3.4 and we look at the cross. We start with the parent generation of homozygous round. They tell us it's homozygous, times homozygous wrinkled. And we get the F1 all around. So that tells us that round is dominant and wrinkled is recessive, and the F1, so this is the F1, these are the parents, the F1s are all heterozygous. So if an F1 plant is back-crossed to the parent with round seeds, so back-crossed means it's back-crossed to one of the parents in the previous generation. And a back cross could be to either one of the parents. So in this particular case, it's going back to the homozygous dominant parent, and so you're going to get both of these. The only thing you can get from that parent is a big R. If you had back cross to this one, it would be a test cross and you would get 1 to 1. But they're asking about this one. So it should be, that's a question. What proportion of the progeny will be wrinkled? Well, we go big R across to big R, little r. They're all big R something, so they're all round. Alright, so it should be 0. Oh, let's go back. Alright, so it was 0. So that makes sense. Simple enough.
Doing genetics, we really like to think in terms of probabilities. Instead of simply a 1 to 2 to 1 ratio or a 3 to 1 ratio, we would think of 1/4 to 2/4 to 1/4 or 3/4 to 1/4. Anyway, if we put our ratios into fractions, you can think of those as probabilities of that particular outcome occurring, and then we can apply that to predicting the outcome of separate events occurring at the same time. For example, what's the probability of getting 3/4 round plants and getting at the same time getting 3/4 green plants? You could multiply those together to get 9/16. The addition rule is if you have a situation where you say, what's the probability of getting this thing or that thing? And if it's an or question, then you can simply add the two probabilities together. We'll look at some examples of that down the road, but just remember that rule. If it's an and question, the probability of A and B, then multiply the individual probabilities together. If it's the probability of A or B, then add the individual probabilities together.
You have a 1/6 chance that that's going to be a 4. If you roll it again and you want to know what's the probability of getting 2 4s in a row, well, there's a probability of getting a 4 the first time is 1/6. Probability of getting a 4 the second time is 1/6, and the probability of getting them both in a row is 1/6 times 1/6. However, if you roll all 6 die in that particular roll, you want to know what's the probability of getting either a 3 or a 4. The probability of getting a 3 is 1/6. The probability of getting a 4 is 1/6. And so the probability of getting one or the other is the addition of those two probabilities. So 1/6 plus 2/6 equals 1/6 plus 1/6 equals 2/6, which is 1/3. So those are just keep those in mind. If you say and, then multiply the probabilities. If the question can be phrased as an or situation, then you add the probabilities. So in this case, if the probability of being blood type A is 1/8 and the probability of blood type O is being 1/2, what's the probability of being either blood type A or blood type O? Well, in this case, you add them together because it's like one flip of a coin. It's one situation or rolling one die, and the probability of one outcome is 1/8. The probability of the other one is 1/2, and so 1/8 plus 1/2 is, well, 1/2 is 4/8, so it should be 5/8. And so that's what it is. So in this case, you can't say what's the probability of being type A and type O with one child, because you can't be both. It's got to be one or the other. So we use this, of course, a lot in looking at basic genetics. And so if you do a particular cross, and in that cross you have a probability of 3/4 that you're going to get a tall plant, and you have a 1/4 probability of getting a green plant, what's the probability of getting a tall and green plant? You simply say 3/4 probability of getting tall, 1/4 probability of getting green, 3/16 getting both at the same time. So we use that a lot. We use that, you might recognize this from dihybrid crosses. You'll see that where we get 9/3/3/1, that's really 9/16, 3/16, 3/16, 1/16, and we can get that by just combining probabilities of different events. And it's much quicker than doing Punnett squares. So let me get this out of the way. So I wrote that down. So tall and green is 3/16. All right, and we talked about the test cross a little bit and ratios and simple crosses. So let's just take a quick look at these. So a test cross is always something crossed to something else that is homozygous recessive. So it does not have to always be a heterozygote being crossed to something else, but that's usually what it is. So a typical test cross for one gene always gives you 1-to-1 ratio. A test cross for 2 genes gives you 4 possible outcomes in this particular ratio. And then I just threw in there, for the heck of it, if you have 3 genes and you do a test cross, the main thing here is just that the test cross is homozygous recessive for all 3 genes. We can look at where these come from, but these are pretty standard crosses that you want to get reasonably familiar with. So obviously, or I hate saying obviously, but as you know, or you're familiar with, big A, if you have a monohybrid, it can segregate like that. And if you do a test cross, you can only get that from the other parent. And you can only get these outcomes. So if you get this big allele, you get this genotype. If you get this little allele, you get this genotype. Similarly, if you have this, you can get big A and big B together, big A and little b together, little a and big B together, little a and little b together. So you get very used to doing that. These are the gametes. And these are the gametes that you get from a dihybrid. And these are the gametes that you get from a monohybrid. Now this isn't a monohybrid cross because we're not crossing it to a monohybrid, but this by itself is a monohybrid. And this is the types of gametes you get from a monohybrid. If you do a test cross, you get this result. If you had done a monohybrid cross, you would have got the other result. Same here, dihybrid gives you these gametes, but if you do a test cross, then the offspring that you get look just like this. And that's due to independent assortment that we'll get to in a minute. But it's really good to just sort of get used to specific ratios. So if you see a 3-to-1 ratio, think monohybrid cross. And then you can write the genotypes like this. If you see 1-to-1, well, that sounds like a test cross involving one gene. Gives you that. Uniform progeny, this is just if you ever cross anything to something that's homozygous dominant, then you're going to get all homozygous dominant, or at least all dominant. So here, if anytime you have one of these things is homozygous dominant, you're going to get all dominant offspring. Now, in this case, in these two cases here, the genotypes are a little bit different after the cross. But then another one that comes up a lot is that if you have something that's homozygous recessive, and you cross 2 of those together, the only thing you can get out is homozygous recessive. So these are just common things that you should look for. At the genotypic level, if you have a monohybrid cross, you get a 1 to 2-to-1 ratio. If you have a monohybrid test cross, test cross, you get a 1-to-1 ratio. And I don't know what you call this thing. Uniform progeny, same kind of deal. So let's move on to dihybrid crosses. So dihybrid cross means that you're dealing with 2 genes, or 2 traits, or both. And so now we just have to keep track of 2 genes at the same time. But the best way to do that is just to think about each gene independently. So what can we get from this cross? Well, we can get a big R and we can get a little r. What can we get from this cross? We can get a big Y and a little y. So let's cross these together. What we can get from here, what's going on, is we have meiosis. And then what independent assortment means is that if you get a big R, you can get a big Y. or if you get a big R, you could also get a little y. So you can get either one of those things. And the same thing with little r. You can get a big Y, or you can get a little y. It's the same thing as the last slide where we said big A, big B, etc. And so, and you can get these all in equal proportion, 1/4, 1/4, 1/4, 1/4. So that's what we have over here. And then if we have the same thing from this cross, that gives you the gametes over here. And then if you fill these in and look at all your different combinations, you get 9/16, 3/16, 3/16, 1/16. And if you look at these things, what we end up with is we get a 9/16 or anything that gets a big R, and then either a big R or a little r. And gets a big Y, and then either a big Y or a little y. So these 9 that are smooth and yellow all have that format. So big R, big Y, big Y. So in other words, all these guys have at least one big R and one big Y. All those guys, then this little guy over here by himself. And then these guys, you can have a big R and 2 little y's. So all of those are, if you get this big R, oh, we can't get it there, so where do we see some little y's? Here's a little y, here's a little y. So big R, well, you just have to look through here and look at those things where you get at least one big R, and then you get 2 little y's. So where's that? Here's one here. One big R, 2 little y's. And those guys are round and what are 2 little, and green, obviously. I'm looking at it green. These guys are wrinkled and yellow. Sorry, wrinkled, big Y, yep, big Y is yellow, and little y is green. And so you get those. So that's where you get the 9 to 3 to 3 to 1 ratio. Looking up, you can do your Punnett square, and here's your phenotypes down here. That means independent assortment. What that means, the fact that these guys are all equally likely, the fact that these guys are all equally likely, that occurs because of independent assortment. So if you see 9 to 3 to 3 to 1, it means that independent assortment has occurred. All right, so that's a couple things. And then the other thing is that this only happens in a dihybrid cross. This is only true if you have 2 dihybrids and cross them together. If you take a dihybrid and do a test cross, you won't get this. If you do anything else, you won't get this. So there's only one time, one type of cross where you get this. Is if you have a dihybrid cross and 2 things, dihybrid cross. So crossing 2 dihybrids together, and we get independent assortment. If those 2 things are true, 9, 3, 3, 1. And there's a close-up of all these different things. There's your 4 different kinds of gametes. Just get used to doing that. Some students get a little tricky as what do you put together. Just look at each gene independently. So big R, little r, let's make those a little bit bigger. Big R, little r, you can only get a big R or a little r. So you can never have 2 R's in the same gamete. And if you have big Y, little y, they segregate away from each other. So you can get either big Y or little y. So get big R or big R, little r, or big Y or little y. But in terms of R's and Y's, they can get together any way they want. That's independent assortment. So how does that work with chromosomes? Well, this doesn't show it as well as I would like it to. At least it doesn't have any crossing over on there. So this is just showing independent assortment. So here's our 2 chromosomes. These are homologous, 2 pairs of chromosomes, and these are homologous. This is DNA replication, or S phase, where the chromatids get replicated. And you'll notice that all of the sister chromatids are identical. So big R is identical, little r, etc. And then it shows them lining up in metaphase. But they don't really line up like that in metaphase. They line up like this in metaphase. I'll just draw my chromosomes as simple X's. they've got the Y chromosome, not the Y chromosome, but the chromosome with the Y on it, like that. Big R have to be identical. Little r have to be identical. Big Y. Tell you what, I'm actually going to make this a little y over here. Little y, big Y, big Y. Why, you might ask? Did I switch that? Because this is how they're lining up. And when they line up that way, these guys will go this way. And so now little r went with big Y, and big R went with little y. But over here, we'll line these chromosomes a little differently. And we'll line up. Still put big R's over here. It's fine with me. If we have big R over there, little r has to pair up with it. But now we'll put big Y over here and little y over here. Now we've got it lined up so the little guys go together and the big R and big Y go together. So when these things segregate, big R, big Y go together, little r, little y go together, big R, little y go together, little r, little y go together. Then we have these guys line up like this, actually, like it's mitosis. And we can have a big R here and a big R here and a little y here and a little y here. Then we do meiosis 2, and big R goes with little y, and that's what we've got here. So this is independent assortment. The fact that that can happen or that can happen. A lot of scribbling, but independent assortment is possible when genes are on different chromosomes. The way they line up during metaphase 1 makes it so that the alleles of one gene can assort independently with the alleles of another gene. So we really like to apply probability to any kind of genetic cross if we can. Punnett squares are good for illustrating concepts, showing you how alleles go together. But if we're going to predict the outcomes of crosses, and we want to do that relatively quickly, it is much better to do combined probabilities, figure out the probability of one thing, figure out the probability of the other thing, and then multiply them together to see what happens. All right, so let's take a look at that. So we'll combine some probabilities. And they also call this thing a branch diagram, which we sort of use. All right, so this isn't too bad. So there's our dihybrid cross. And essentially all they're doing is looking at each gene independently. And it's just a super good way to do it. Anytime you have a big cross, look at each gene independently. So big R, little r is a dihybrid cross. Overall it's a dihybrid cross. But individually, there are two monohybrid crosses. So big R, little r, cross to big R, little r. You get this, 3/4, 1/4. So that's this. Big Y, little y, cross to big Y, little y. You get 3/4 big Y, 1/4 little y, little y. So then what's the probability of getting these combinations together? So this branch diagram business is saying if you have 3/4 that are round, of those, 3/4 of those are going to be yellow, and 1/4 of those are going to be green. So to figure out how many are going to be both round and yellow, you multiply these things together. 3/4 times 3/4 is 9/16. How many are going to be round and green? Multiply this one by this one. 3/4 times 1/4 is 3/16. Round green. Wrinkled is 1/4. Yellow is 3/4. Multiply those together, you get 3/16, which is wrinkled and yellow. And then 1/16. If you have 1/4 probability of getting wrinkled, 1/4 probability of getting green, you get 1/16 probability of getting wrinkled angry. And these genotypes are like this. And there you have it. So practice that. That's the same thing. Just to point out that it's easy to use this principle to predict the outcomes of any cross. Now here we wouldn't want to list all possibilities. So this is really a tri-hybrid cross. Now we could, if we wanted to, pick predict all possible outcomes. But let's just pick one possible outcome. What's the probability of getting that? So probability of getting the dominant A phenotype, the dominant B phenotype, and the recessive C phenotype, whatever they are. What's the probability of getting those? Well, we know that a monohybrid cross for big A gives you a 3/4 probability for this. So we expect 3/4 for this. Same thing for B, because B is also a monohybrid cross. C is also a monohybrid cross, but we're asking for 2 little c's, and we know there's 1/4 probability of getting that. So the probability of getting all 3 of them at the same time is 3 times 3 is 9, times 1 is 9. 4 times 4 times 4 is 64. So that's the combined probability. The fact that there's 64 down here means that if you did a Punnett square, means you're going to have 1, 2, 3, 4, 5, 6, 7, 8 possibilities on this side, 1, 2, 3, 4, 5, 6, 7, 8 possibilities on this side, and 64 possibilities inside this humongoid Punnett square. So rather than doing a big old humongoid Punnett square and then going to count up all the ones that look like this, we just figure out the probability of each and multiply them together. And that's what Mendel did. Mendel did not. So we'll stop this video there. That's the basics of monohybrid crosses, test crosses, dihybrid crosses, a little bit of how chromosomes explain independent assortment and the types of combinations you get when you have a dihybrid and you look at the different kinds of gametes you can get. The second half of the chapter, the next bit, is about chi-square and binomial, and we'll put those on their own video so that this one isn't any longer than it has to be. So that's it for now, and if you want to look at the chi-square and binomial, take a look at that one. All right, see you later.